Hello dear students, welcome back again to Chinmoy's YouTube channel. So in yesterday's episode, we have already spoken about what is transpiration, what is evaporation, what is the difference between transpiration and evaporation, what are the three main types of transpiration and the mechanism of stomatal transpiration, cuticular transpiration and lenticular transpiration. So today is a brand new episode. So we are going to discuss today about the experiments on which we can prove that whether transpiration is happening or not in plants. So how it is happening and different many 4 to 5 experiments we are going to discuss today, as well as in the weighing method how transpiration we can discuss in Ganong's potometer and all we will discuss today. So let us start with today's episode of transpiration part 2. So what are the learning objectives of today's lesson? So at the end of this lesson, we will be knowing that experiments to show that water vapor is given out during transpiration, to demonstrate the transpiration in plants with cobalt chloride paper, and to show that more transpiration occurs from the lower surface of the leaves. So this is the first experiment which will just show that whether we are telling transpiration is happening in plants or not, we are proving with an experiment that transpiration is happening in plants. Now the second experiment will demonstrate that transpiration is happening in plants with the help of a special cobalt chloride paper. So we will see the uses of cobalt chloride paper. Now the third one is to show more transpiration occurs from the lower surface of the leaves. So in the lower surface of the leaves, we know that stomatal pores are more than the upper surface of the leaf. So this we will prove with the help of an experiment that transpiration occurs more in the lower surface of the leaves. Now the next one is two methods of measuring the rate of transpiration. So how fast or how slow transpiration is happening with the help of weighing method or with the help of Ganong's potometer. So we are going to learn today all these 5 to 6 experiments. So if you want to know more regarding transpiration, you need to subscribe to my channel to stay updated and to stay tuned to all my posts which I am updating regularly. Please do subscribe to my channel and don't forget to like and share my videos with your friends also, so that they can also study these same lessons in a systematic manner as I am teaching you. So please do write in my comment box what are your queries, so that in the next episode I can clear up with the answers. Okay, so let us start with the first experiment. Now here we are with the first experiment to show that water vapor is given out during transpiration. So in this diagram what we can see that there is a potted plant inside and it is covered by a bell jar. Okay, and we have already covered this potted plant with a polythene bag tightly tied around the neck so that no evaporation should take place from the soil part. Okay, and we have also on the sides of the bell jar we have put Vaseline throughout so that no water can enter from the outside. So if we are keeping this potted plant with the inverted bell jar for 2 to 3 hours in sunlight, so we are putting this plant under sunlight for 2 to 3 hours, and we are keeping a similar set of a bell jar only without a potted plant. So if this is A, suppose, and we are taking a control bell jar B and we are keeping that empty bell jar without any plant inside. This is the empty bell jar. The Vaseline is being applied on the sides of this plate also, and no plant is there inside. Okay, no plants are there inside, and here we can see one potted plant is there. So after 2 or 3 hours in sunlight, direct sunlight, if we are keeping it under 2 to 3 hours, what we will observe? So the observation is that after 2 to 3 hours, we can observe that droplets of water vapor on the bell jar. Okay, so we are seeing that droplets of water vapor are being seen in the bell jar, which is due to transpiration. So the water is lost from the aerial part of the plant. So we have already covered this potted plant, so no evaporation happened from the soil, from the pot. So only from the aerial parts of the plant the water is evaporated or the vapor has come out. So this proves that water vapor is given out during transpiration only. But in this plant you can see, in this one there you can see that this is the control jar. So where no plant was kept, but this bell jar was kept in the similar condition 2 to 3 hours under sunlight. But no evaporation happened here because no plants is there. Nothing to evaporate or no transcription can happen here as there is no plant inside. So it is proved that water vapor is given out during transpiration. So this one is the first experiment. Now we will move on to the next one. So this is a very easy experiment to just prove that water vapor is given out during transpiration. So you should know the label diagram and with all the labelings, and a neat and clean diagram should be given. So first you should write the aim of the experiment, then the procedure, then the observation, and the materials required also should be mentioned when you are writing the experiment. So you write it down in steps, and with a diagram you should take two plants. One is A, one other is B. One is control where there is no plant, and another is the experimental setup where we are keeping the plant. Okay, so now we will move on to the next one. Is it clear to all? Okay, we are moving to the next experiment. Now here is the second set of experiment, that is to demonstrate that transpiration is happening in plants with the help of cobalt chloride paper. So we have taken here 3 bell jars. So in the first we have taken a potted plant but no cobalt chloride paper. In the second one we have kept a potted plant with a cobalt chloride paper, but in the setup C there are no plant but only a cobalt chloride paper is kept. So what is the characteristic of cobalt chloride paper? Cobalt chloride paper, it generally is blue in color, but it turns pink. So it turns pink when it is absorbing a huge amount of moisture or slight amount of moisture in it. So let us see what is happening in this experiment. So first we have taken a potted plant, we have covered the full base as in the earlier previous one also with a plastic covering so that no evaporation occurs from the soil. In the second one also same thing is happening, that we are not allowing any evaporation to happen from the pot or the base of the plant, and here no question of plant is there, only the cobalt chloride paper is kept. So we are keeping these potted plants for 2 to 3 hours in the sunlight. Okay, under sunlight it is kept for 2 to 3 hours. So what is happening? What is the observation after 2 to 3 hours? In the first plant, in the first one, we can see the droplets of water vapor are there throughout the bell jar. Okay, so these droplets are condensing and droplets of water vapor are present. Now in the second one we can see that these droplets of water vapor are there, as well as this paper, it is turning pink in color. So this is turning pink in color, that cobalt chloride paper has absorbed some amount of moisture, which is due to the transpiration which has happened in these plants after being kept for 2 to 3 hours under sunlight. So this is happening for the second plant, and here as no plant is there, no question of moisture generation is there, no condensation of moisture is there. So this paper, the same color blue, it remains blue only. So there is no change in the color of this cobalt chloride paper. So hence it is observed from the experiment that cobalt chloride paper, which is generally in dry state blue in color. So cobalt chloride paper when it is dry, it is blue in color. It is blue in color. So in the second one, when it is absorbing moisture, it is turning to pink. So this is the pink color. So this is absorbing moisture, it turns to pink. Okay, it is turning pink. So this is how we can prove that during transpiration water vapor is produced, and that water vapor is condensed in the form of moisture which is being absorbed by the cobalt chloride paper. So hence we have proved that presence of moisture or water vapor due to transpiration of plants only. So it is due to transpiration. So is it clear? If you are having any queries, you can ask in my comment box also. I'll clear up in the next episode. So let us move on to the next experiment. Now here is the third experiment where we are proving that more transpiration is occurring from the lower surface of the leaves. So this experiment also we are taking cobalt chloride paper, as it is a good indicator of the presence of moisture during transpiration, and it shows a drastic color change. So we have told in the previous slide also, it is generally blue in color, but after absorption of water vapor it turns, or moisture, it turns pink. So what we have done here, we have taken leaves of either balsam or China rose plant. What we are doing, we have taken these two leaves and we have taken these leaves should be attached to the plants. You should not detach the leaves from the plants. Okay, so what we have done, we have taken a glass slide. In this too you can see a glass slide has been taken, and to that we have stuck cobalt chloride paper. So cobalt chloride papers is stuck. So one slide with the cobalt chloride paper touching the leaves, we are putting it with the gems clip on the surface of the upper surface of the leaf, and another one we are putting in the lower surface of the leaf. So this one, suppose this is the upper surface of the leaf, this is the lower surface of the leaf. Okay, so this is how we are putting gems clip on the end of two glass slides, which we have inside. We have kept the cobalt chloride paper. So one we have gems clipped on the upper side of the leaf and another on the lower side of the leaf. So we have left it in the open sunlight for 2-3 hours, as these leaves are already attached to the plants itself. So there is no question of attaching them or taking the potted plant outside, inside, nothing. So you have kept the pot already in the sunlight for 2 to 3 hours. So after 2 to 3 hours, what is happening? The thing what is happening is, we can see the observation that the cobalt chloride paper, which was being attached to the upper side of the leaves, is slowly turning to pink, whereas which was on the lower side of the leaf has already drastically turned to pink color. So hence it is proved that transpiration occurs from the lower surface much faster than from the upper surface, because we have already studied about the structure of stomata in the previous lesson. I have told you when I taught you turgidity and flaccidity and the turgor pressure, I have already taught you the structure of the stomata and where are they located. So we have seen in the lower part of the leaves only they are located more. So here from this we can prove that transpiration occurs from the lower surface of the leaves more, as number of stomata is more on the lower surface of the leaves, and so the cobalt chloride paper, which turns more pink in the part where it was attached or it was stuck to the lower side of the leaf, whereas which was stuck to the upper side, it is gradually turning to pink, but it is taking much longer time than which was attached to the lower surface of the leaves. So hence it is proved that transpiration occurs from the lower surface of the leaves more. Is it clear? So now we are moving to the next experiment. Now here we are discussing about the rate of transpiration with the help of weighing balance. So we have taken a weighing balance here. So what are the materials required? We have taken a weighing balance here. We have taken two test tubes. In one test tube, which is the experimental one, we have put one twig or one external part of a plant. We have taken a plant part, okay, and in this one we have not kept anything, so it's a control one. So this is the experiment is happening here, and this is the control part, which with that we are going to compare this A. So A and B are the two setups here. So this test tube, here we have put oil on the surface of this water. Why? Because we want to prevent the loss of water from the surface of this water, because this is evaporation, this is not transpiration. So we have told the difference between evaporation and transpiration in the previous episode. So if you have not yet gone through that episode, I am putting the link above. We have already spoken what is transpiration, what is evaporation, and what are the main differences between transpiration and evaporation. So here we want to prove transpiration is happening, and there is a weight difference after transpiration has happened. So we don't want evaporation in this setup. So we have put oil on the top of water. So this is water, this is a layer of oil, and here in this control test tube no plant is being kept. But in A there is a twig or an external part of plant is being kept. So after keeping this full setup in under the sunlight for 2 to 3 hours, what is happening? We will see that gradually the weight of this A reduces. Okay, so the weight of A reduces. So why it is reducing? Because the water is lost due to transpiration. So water is lost due to transpiration, and there is a loss of weight of this plant due to transpiration. So loss of weight is happening due to transpiration, whereas this weight remains stagnant. There is no loss of weight in this control test tube or in this B, because there is no plant and there is no loss of water from the aerial parts of the plant. And we have already stopped the evaporation here because we have put oil, so no evaporation from the water surface also. So transpiration causes the loss of weight in this A plant, whereas no loss of weight is happening in the B. So this proves that transpiration is happening, and the rate of transpiration is in the first one reduces the weight, and that is why it is done in this weighing balance method to prove. So hence we have proved this experiment also. So now we will move on to the next one. Now here it is the second experiment where we are measuring the rate of transpiration with the help of weighing balance. Now here weighing balance, you can take a weighing balance here. Suppose it is a weighing balance. Now what we are doing, we are first taking a plant and we are inserting the plant in this in this container and we are filling it with water. Okay, it is filled with water. And what we are doing, we are taking a graduated tube next to it, which is also, this water is only, it is here up to a level, there is this water is there. Okay, so we are keeping this full setup in sunlight for 2 to 3 hours. So what is happening when the water is being evaporated from this particular vessel? So gradually the water from this vessel, this graduated tube, is slowly passing to this container. So what is happening then? This level of water is falling. So we can just mark the difference in the water level. So suppose it was here, gradually after transpiration for 2 to 3 hours, the level has come down to this. So in this graduated side tube also we can mark the level of water, how much it has come down after 2 to 3 hours. And as we are inserting a plant inside this plant, in this pot, we can take the measurement of this full setup in a separate weighing balance also. So what will happen there? We will see before starting the experiment the weight, what was the weight, and after completion of 2-3 hours of the experiment we can see that the weight is reduced a lot. So why the weight is reduced? Due to loss of water from the aerial parts of the plant. Now here the water also, it has been absorbed here, and there is a constant loss, it's keeping on happening, a constant loss of water is happening from the aerial parts of the plant. And this, after weighing also, we can prove that the weight beforehand was more than that afterwards. So hence it is proving that the rate of transpiration reduces the weight of the plant. Okay, so because the water is being lost from the aerial parts of the plant, so the weight of the plant which was taken before the experiment started was more than the weight of the experiment which has been taken after the experiment is over. So this is clear to you all, right? So we are moving on to the last experiment, that is the Gannong's potometer. Now here is the last and the most important experiment here, to measure the volume of water uptake by the plant during the transpiration using potometer method. So this is Gannong's potometer. So what are the things here? So potometer, such as Gannong's potometer, which I have taken here, it is a device which is made up of glass, okay, and it is used to measure the rate of transpiration by a leafy shoot. So what is this plant? This is, we are generally taking the Plectranthus plant, okay, it is the Plectranthus plants, and a twig of this plant has been cut and placed in this tube. Now here we have taken a reservoir. So why this reservoir has been taken? So that we can give it a constant supply of water when it is required. So no need to stop the experiment and fill in water. Now here we have taken a beaker of water which is generally pink in color, so that when the transpiration water loss is happening here, the water, when it flows from this part, we can see that how much water is being replaced by this colored water. Okay, now what is done here, there is a capillary tube here, you can see, and we can see this volume scale over here. Now what we have done here, we have generally introduced an air bubble. So an air bubble has been introduced in this horizontal line. So air bubble has been introduced in the capillary tube by slightly lifting the capillary tube. So this is a very difficult process. If you are holding the entrance of the capillary tube with your fingertip only, just slightly removing the fingertip, you can insert only one bubble is required. So what is we are doing? We are lifting that, we are dipping that in the beaker which is containing the colored water, and as the shoot is transpiring, it is constantly transpiring the water, it is constantly transpiring, and the vapor lost, which causes the water to be drawn inside the capillary tube, through the capillary tube, and the colored water is entering the capillary tube through this, and the movement of bubble has also been seen. So with the entrance of this colored water in the capillary tube, as the bubble is moving, it also moves through the capillary tube, and thus the reading of the capillary tube is changing. So bubble is moving, water is moving from this, as the water is transpiring from here. So the water lost is being compensated by the water which is drawn from the beaker, colored water which is drawn from the beaker through the capillary tube. So this is causing the movement of the air bubble also, as well as the water inside the capillary tube also. So the reading in the capillary tube gives us the volume of water which is being lost in the given duration of time. So suppose we are keeping this setup for 2 to 3 hours in the sunlight. So in 2 to 3 hours we can say that this volume of water, that is the marking on the capillary tube which has been recorded. So that volume of water has been replaced, or that amount of water has been evaporated in that time. So this is Gannong's potometer. It is a difficult experiment to perform, but though we can say that this is a difficult experiment which is measuring the volume of the water which has been replaced during transpiration, and there are several limitations. So this is a very important thing. What are the limitations of this experiment? So we will know the limitations now. Now, there are few limitations of this Gannong's potometer. Now, in the previous slide, when we are discussing about this experiment, we should mention the main fact that when we are inserting the twig with this rubber stopper and this stopcock, we should see to it that no air bubble enters this water. So first we are inserting this twig inside with a rubber cork, and then we are inserting water into this tube. Okay, so no air bubble should enter because we are inserting another air bubble from this beaker, right? So it is being introduced in this capillary tube. So it should be noted that while filling in this total setup, no air bubble should enter into that setup. So this apparatus should be filled, ensuring that no air spaces are there in between. So what are the limitations, or what are the limitations of this Gannong's potometer experiment? This is a very important question which is generally coming in your exams. So it is very difficult to introduce air inside the capillary tube. The twig does not remain alive for a long time, and any type of external temperature may affect the position of the air bubble. So suppose the temperature is very windy, very stormy outside, so air bubble in the capillary tube can get disturbed. So it's just a mere bubble. It is very delicate air bubble. It can get disturbed with the external temperature changes, or whether if the temperature is very hot, if there is too windy, or the air velocity is too much more, so it will affect the position of the bubble in the capillary tube. So these are all the main experiments of this transpiration lesson. So if you want to know more about the factors which are affecting transpiration, what is the significance of transpiration, and how our plants are being adapted to reduce the excessive transpiration. So this I'll be coming in the next episode. So you need to subscribe to my channel to learn more about this transpiration, and I'm coming with more lessons for your 10th ICSE biology as per your ICSE curriculum. So please stay tuned with all my lessons, all my updates, so that you can get to know whatever I'm posting. So don't forget to subscribe to my channel. Stay tuned, stay updated. Thank you.